Hello, and thank you for joining us for this Capsugel Dosage Form Solutions webinar entitled Hot Melt Extrusion of Amorphous Solid Dispersions for Bioavailability Enhancement. This is the fourth webinar in our Bioavailability Enhancement series. My name is David Vodak, and I am the Vice President of Chemistry at Bend Research, part of Capsugel's Dosage Form Solutions, or DFS unit. Over the next 45 minutes, Dr. Aaron Goodwin will discuss the application of amorphous solid dispersions via hot melt extrusion processing in preclinical formulation identification, including stability and performance considerations. We will provide case studies to demonstrate the technology's range of application and the resultant solubility improvement achieved. We will then have about 10 minutes to answer any questions from you. When you have a question, please submit them at any time using the Ask a Question tab on the left of the screen. Feel free to use the tab at any time. We will answer your questions at the end. And if we don't get to your question, we will follow up directly with you. Finally, at the end of the webinar, we will have a brief survey. We thank you in advance for your participation and look forward to your questions, comments, and suggestions. This all helps to make our offering of these webinars more relevant to what the scientists are actually trying to learn about. I'd like to introduce Dr. Aaron Goodwin. Dr. Goodwin holds a PhD in chemical engineering from Oregon State University. His areas of expertise are technology development for drug solubilization, which includes formulation and process development for hot melt extrusion, attrition milling and precipitation of nanocrystals, and formulation development of oral solid dosage forms. Okay, and with that, I'd like to get started on a brief introduction of our company. Bend Research is now part of Capsugel's Dosage Form Solutions business unit, headquartered in Morristown, New Jersey, providing access to additional technologies, research and development capabilities, and global manufacturing infrastructure for the advancement of client compounds from concept through clinical to commercial manufacture. And here just a brief bit of information about Bend Research. We are a leading scientific research and development and manufacturing company located in Bend, Oregon, and founded in 1975, and currently have about 250 employees. Our mission is to improve the standard of living through the development and application of the best new technologies. Some of our key values listed here include curiosity, science and engineering excellence and the reputation to maintain that, quality culture, reliability, focus, honesty and direct communication, family, loyalty to our customers, and of course the one that makes people smile, no whining. Capsugel's DFS business utilizes an array of proprietary technologies and specialized manufacturing capabilities to solve customers' most pressing product development challenges, including bioavailability enhancement, modified release, abuse deterrence, biotherapeutic and formulation and processing, and inhalation formulation and processing. The company's fast-to-clinic program streamlines product development from pre-formulation through clinical and commercial supply for finished dosage forms. As shown in this slide, we think of our technologies, the tools in our toolkit in these three pillars, and all of these technologies, based on a customer's problem statement, can be used to brought to bear as a function of the target product profile, customer therapeutic target knowledge, deep formulation science and understanding, the process development engineering, and the market drivers, and that all with these technologies and the expertise to use them is brought together to deliver on products. So that's enough of the brief introduction, and let's get to the science. So with that, I'd like to turn it over to Aaron, and let's learn about hot melt extrusion. Thanks, Aaron.
Well, thank you, David. Thank you for the introduction. So this slide kind of sets up the problem statement and shows the developability classification system. And what we're seeing now is more and more compounds in discovery and development are DCS class 2. And what that means is that their bioavailability is either limited by a poor dissolution rate or poor solubility. In fact, this makes up about 30% of the currently marketed products, but makes up about 60 to 70% of the products in development. And BRI has many capabilities and many technologies that we use to address these problems. So this next slide shows our conceptual bioavailability enhancement technology applicability map. Now this plot is wrong, but it illustrates the way Venn Research approaches technology selection for bioavailability enhancement. Based on our experience, crystalline salts, amorphous, and lipid-based technologies are the most widely applicable, and there are a large overlap in these technology approaches. Typically, it's a dimension that's not really shown on this simple plot that further drives the technology selection. For example, target PK profile, metabolism, permeation, and finally dose. One solution for bioavailability enhancement is to make the API amorphous. And the amorphous form of the API or the drug has a higher free energy and therefore has a higher solubility and dissolution rate. And this is because there's no long-range order like in crystalline drugs. And this form can either be stabilized thermodynamically or kinetically. So one way to do this is to make a solid dispersion or amorphous solid dispersion. And two technologies used to manufacture these amorphous solid dispersions are hot melt extrusion and spray dried dispersion. So what this slide shows is just a comparison of the two technologies and some advantages and disadvantages for both. So in general, hot melt extrusion is a continuous process that has a lower capital and manufacturing cost. However, the formulation is limited to miscible systems at the process temperature, whereas spray drying the formulation is less constrained. Just to compare some of the powder properties of the resulting drug intermediate is HME solid dispersions tend to have excellent flow, which may enable direct compression in a lot of cases. Now, spray-dried dispersions tend to have very good powder compressibility, but often dry granulation is necessary to improve powder handling downstream. Now, HME is a very well understood and mature technology that has resulted in a variety of currently marketed products. Some of the marketed technologies include dispersions, granulations, and shape systems. And really, the roots of hot melt extrusion are in shape systems. However, it's being used more and more for solid dispersion technology. Posaconazole, an antifungal, has recently been approved by the FDA. And Venn Research's first pharma-related project, highlighted here in the gray bar, is a melt congeal, a type of HME. And Venn Research developed this with Pfizer. So this slide shows a typical hot melt extrusion process train. And there are a lot of permutations to this, but this is a very simple, simplistic way to look at this. And in general, what we would do is you have a blend, and that's a polymer and a drug in the simplest of cases. And that's fed either gravimetrically or volumetrically to an extruder. And then the extrudate is calendared, cooled, and pelletized, and then finally milled. This is a very robust and highly reproducible steady-state operation. It's very easy to scale up, and this will be discussed later in the presentation. It also allows for flexible batch size and continuous operation, and this has the potential even through tableting or encapsulation. Now, at the heart of this process train is the extruder. And a twin-screw co-rotating fuller intermeshing extruder is preferred for hot melt extrusion. And this is simply because it has better mixing than a single screw or non-intermeshing extruder. The twin screw extruder consists of 2 rotating screws and a segmented barrel. Some of the process parameters are listed on this slide. So the continuous independent process parameters, and by continuous I mean they can be changed on the fly, are screw speed, feed rate, and barrel temperature. Some non-continuous variables are screw design, barrel design, and die design. And then finally, some of the dependent variables are the melt temperature, the residence time, the downstream processability of the extrudate, and the dispersion quality. Now what makes extrusion so versatile is that you can have multiple unit operations in a single extruder. The barrels are heated and cooled independently, and the screw has removable elements. So what this allows us to do is to design a barrel temperature profile and a screw design that allows us to achieve our goal, in this case, manufacturing amorphous solid dispersion. So on this slide, we show a typical setup for our screw design and for our barrel design. And typically, HME has about 5 unit operations. The first one is feeding, and this takes place in the first part of the screw. Now the polymer and API can be introduced independently or as a blend. And this is done volumetrically or gravimetrically, and the powder enters via feed throat, as seen in the schematic. The feed barrel is actively cooled to prevent low melting point material from sticking. And now the next unit operation is conveying and melting, and this occurs after the feed zone. Now the primary purpose here is to move powder away from the feeds, and energy is introduced via heat transfer from the blocks and shear. And this is dependent on the melt point of the components as well as the particle size of the components. So the third unit operation is mixing. And here our goal is a well-mixed material at the molecular level, which creates a single phase. Now the amount of mixing depends on if we're dissolving the API versus melting the API into the polymer matrix. And the 2 types of mixing, as seen on the slide, are dispersive mixing and distributive mixing. So dispersive mixing is the more intense mixing, and this takes place, we use something called kneading blocks to do this. Now the larger the angle, the more dispersive mixing. So a 90-degree kneading block is all dispersive mixing, and there's very little conveying, whereas a 30-degree kneading block, there's more conveying and less dispersive mixing. Now, there's a cartoon in the middle that kind of shows the difference between the 2 mixing regimes. As we can see for dispersive mixing, we're taking the black dots in the top of the diamond, and we're further breaking those down to very small domains. Whereas in the distributive mixing, we're just distributing those equally within the space. Now, distributive mixing, we will use combing elements, and those can be seen on the right-hand side of the slide. Also, as alluded to earlier, the small angle kneading blocks have a lot of distributive mixing as well. Now, generally for HME, we will do distributive mixing followed by dispersive mixing, and if we look at the screw design, we can see this. In the first mixing section of the screw, there's 30-degree kneading blocks, so that's a lot of distributive mixing and a little dispersive mixing. And then later on in the screw, we have 60-degree kneading blocks, which is a lot more dispersive mixing and a lot less distributed mixing. Now, for a fixed screw design, the mixing intensity is a function of residence time, specifically the span of the residence time, and screw speed. So faster screw speed is better mixing. Now the 4th unit operation is venting. And here the purpose is simple. It's just to remove residual moisture and gas or air entrained in the powders. And a vacuum can be used to assist. And finally, the 5th unit operation is extruding. And extruding the dispersion out of the dye. And as you can see at the end of the screw, we have special pumping screw elements used to create pressure to flow the extrudate out of a shaped die. Now this slide shows the extrusion equipment available at Bend. So Bend uses twin screw co-rotating extruders, and in our development facility and GMP facility, we have 18 millimeter light strits and a 27 millimeter light strits. We also, in our development facility, have a 19 millimeter BMP. Now we also have a variety of techniques we use for process analytical technology. We have Raman and NIR, and if you look at the picture with the 18 millimeter light strits, you can see there's a Raman probe attached into the die. And now on the picture on the top right shows a variety of Bend Research's dies. We have a large selection of dies, and we have and a lot of these are prefabricated and custom depending on the application and need. So this slide shows our ancillary equipment used for the extrusion process and some of our milling equipment. First is our Sandvik chilled rolls and cooling belt, and this is what we prefer to use for downstream processing. It has a 5-ton and 3.4-ton cooling capacity, and for those that aren't familiar with tons of cooling capacity, that's 1 ton. A ton is a heat of fusion absorbed by melting 1 ton of ice at 0 degrees C in 24 hours. Kind of funny, but it's equivalent to approximately 3,500 watts. And this can be used with any of our extruders. Now once the extrudate goes through the chilled rolls and is flattened and cooled, it's directly fed into one of our pelletizers. And the pelletizer is not pictured. We also have several K-tron loss and weight feeders, and several of these can be used during development to fast prototype formulations if the powders are not blended and fed in independently to the extruder. We also have some high pressure ISCO syringe pumps for liquid injection, and we have a variety of milling, mills and milling equipment. What can be seen at the bottom of the slide is our FITS mill, and we have an M5A and an L1A, and we have a variety of co-mills used in development and GMP. This slide shows Bend Research's approach to formulating amorphous solid dispersions. And really what we're doing here is, this is all about performance. It's all about bioavailability enhancement. And what we need to balance here is the performance and the manufacturability and the stability. And specifically what we want to do is we want to hit our target PK profile, and usually that will be either by increasing dissolution rate or enhancing solubility. But now the formulation has to be manufacturable, meaning we have to be able to scale it and we have to be able to reproduce it. And finally, the drug intermediate needs to be physically and chemically stable. And this is all a function of formulation and equipment configuration and processing parameters. And so we will typically use a 3-step process to define the formulation and target the desired product attributes. We will identify and understand the physics within the extruder, use models and experiments to quantify this physics, and finally we organize all of these steps into a flowchart to help us think more fundamentally about the process. So this shows Bend Research's formulation and process development flowchart, specifically for amorphous solid dispersions by hot melt extrusion. And this begins with technology selection in the middle left. And this begins when an API needs bioavailability enhancement, either to progress a molecule or simply gain exposure for tox. In large, as stated earlier, this is based on the molecular properties of the API and the specific problem statement. If hot melt extrusion of an amorphous solid dispersion is selected as the appropriate technology, we start by defining the formulation selection and the formulation operating space. So our approach to a formulation is to start with a binary system, meaning an API and a polymer, and add additional excipients as necessary to improve processing or in vivo performance. Now, formulation selection is based on matching that target PK profile and stabilizing the amorphous form of the drug to achieve an acceptable shelf life and to address any known chemical stability issues. And this is because the amorphous form of the drug, in many cases, degrades easier than the crystalline form. And we use several tools to narrow down polymer selection and drug loading. Typically what we do is we prototype formulations by spray drying small-scale STDs. And we will use 10 to 100 milligrams of API per formulation. And what we'll look for is solubility enhancement and sustainability by doing in vitro testing. We will also do thermal testing and chemical stability testing by forced degradation. So this slide shows some of the questions we begin to ask ourselves during formulation selection. Now the first question is, does the drug have solubility in the polymer at the processing temperature? And what we're really getting after here is, what is the maximum drug loading versus process temperature we can use, and how sensitive is that going to be to a quench rate? So we first start attacking this question by using solubility parameters. And solubility parameters can be used to predict the miscibility of two substances by determining their individual cohesive energy density. Now, things with similar cohesive energy densities are likely to be miscible. Now, solubility parameters for polar molecules can be calculated according to Hansen, and that's in the upper right. And the way we calculate this, one of the preferred methods we use to calculate this is a group contribution method, and we will typically use the Van Crevelen and, excuse me, I will not pronounce this right, but Hofster's group contribution methods. And we will average those two. And as you can see in the right, these total solubility parameters, denoted delta T, is a function of the individual solubility parameters, the first one being dispersion or van der Waals forces, the second one being intermolecular hydrogen bonding forces, and the third one being intermolecular polar forces. And what we will do is, once we have the solubility parameters for the API and for the polymer, we will rank order polymers for expected solubility at processing conditions. And the closer the solubility, in general, the higher the drug loading that will be possible. So this next slide shows taking this a step further. So this shows the thermodynamics of a drug-polymer dispersion. And we use Flory-Huggins theory to help us determine this thermodynamic relationship. And this is the same model used to predict solvent polymer chemistry. Now, there are many assumptions that go into this model, but it's very pragmatic for its use. And it allows us to optimize drug loading as a function of process temperature. So if we look at the right, there's an equation there, and that's for the delta Gibbs energy of mixing. And there's an entropic term and an enthalpic term in this equation. And where the solubility parameters come in is there is something called an interaction parameter denoted as chi. And chi is derived from the solubility parameters. Now, once we have this relationship, we can plug it in, and as you see on the graph on the left, we have the Gibbs energy of mixing versus fraction of drug. And there's three distinct regions in this graph. The first region is the stable region, and this is where the delta G is negative, and the drug and the polymer will mix spontaneously and will not phase separate. Now the next region is the metastable region, and this is where the free energy is negative, so they will mix, but the slope is positive. So in this region, the solid dispersion will eventually phase separate. And phase separation rate is largely based on diffusion and the activation energy to form a critical nucleus. Now the composition of the phase separated region is constant. Only the size and the distribution change with time. And finally, the last phase is the unstable phase. And this is where the polymer and the drug will not mix, and diffusion is the barrier to phase separation. Composition and size of phase separated regions will change with time. So from this thermodynamic relationship between the drug and the polymer, we can begin to construct a binary phase diagram of the drug and the polymer, and this aids in our drug loading selection. And this is still based on the Flory-Huggins model. And in this particular phase diagram, this is for phenofibrate, a model compound, and PVPVA. The area outside of the binodal line is the stable region we talked about on the last slide. The area in between the binodal and the spinodal line is the metastable region, and the area under the spinodal line is the unstable region. In this case, we used SDDs, and we scanned these using differential scanning calorimetry, and we held these at several temperatures and analyzed the DSC traces to look for phase separations. And the red points indicated phase separation, and the blue points indicated no phase separation. So from this, we can optimize our binary phase diagram. And we can use this to tweak that chi interaction parameter in the Flory-Huggins model. So in this case, we took this information and we extruded the PVPVA and the phenofibrate, and we ran the resulting extrudate through a DSC, and the results are on the right. So what we have is TG versus percent of phenofibrate. And as indicated by the plot on the left, the 10% dispersion, there was no phase separation. And the 25% dispersion, there was also no phase separation. However, at the 50% dispersion, there was phase separation. And now if we look back at the plot at the left, if we go to the 50% one, we can see at the processing temperature, it's predicted that there would be phase separation. So this is a very cool result. It kind of confirms that our binary phase diagram is in fact correct. Now we can take this a step further. Once we have validated our phase diagram, we can take this a step further to determine if quench rate may impact the drug domain size of the solid dispersion. Now, the larger the drug domain size is, the easier it is going to be for the solid dispersion to phase separate. And this is going to affect physical stability. So we have two particular cases here. Case one is the dotted line in the plot on the left, and case two is the dashed line. Now in the dotted line, there is no quench rate effect on drug domain size, and that's because we don't enter the metastable region until below the drug TG. And on the dashed line, there will be effect of quench rate because we enter the metastable region before we hit the TG of the drug. And we have a kind of a conceptual cartoon on the right-hand side. So on the left-hand side, we will we're essentially quenching on the green line. So this is if we're quenching in milliseconds. Really, we never actually enter the immiscible and mobile region. So the drug domain size will be as small as it is at the processing temperature. However, if we look at the dashed line, we start in the miscible region, and as we trace down, we go into the immiscible but mobile region. So in this region, the drug is free to move around and find itself. And the drug domain size here is estimated based on a simple L squared over D, D being the diffusion coefficient calculation. And the longer it sits in this region, the immiscible and mobile region, the larger the drug domain size will be. Once we go below the TG, we enter the immiscible region, and the drug is no longer mobile. So the next question we asked ourselves was, will the solid dispersion have acceptable physical stability at storage conditions? And what we're talking about here is the solid dispersion thermodynamically or kinetically stabilized? And if it is kinetically stabilized, how sensitive to temperature and relative humidity will it be? So the first thing I wanted to talk about is the physical state of the amorphous solid dispersion. Now there's two fundamental issues here, the initial state and the state at infinite time. And so we'll assume that the initial state is the same for each case we're going to talk about here. The thermodynamically stabilized system will have no driving force for phase separation or crystallization. That is, the drug and the polymer are thermodynamically miscible in one another. And on a PXRD, you will see an amorphous halo, and on a DSC, you will see a single TG that changes with composition. For a kinetically stabilized system, this will always change with time. And the kinetic part is how fast will it change with time. So we start with the same homogeneous dispersion, and in the first case, at time equals infinity, for a high TG compound, we will just see phase separation, but not necessarily crystallization. And in a PXRD, we will see this as an amorphous halo. However, in a DSC, we will see two TGs, one for the drug and one for the polymer. And now in the final case for kinetically stabilized system, we have a compound that has a propensity to crystallize. And in the PXRD, we will see an amorphous halo with API diffraction peaks. And in a DSC, we will see a polymer TG and a drug and API melt peak. Now, this is an important factor in determining shelf life and storage conditions for the final dosage form. Now, we often target thermodynamically miscible systems, but we have to settle with kinetically stabilized systems to achieve a higher drug loading. And we at Bend Research have a really good grasp on the physical stability of drug intermediates based on what we call relative mobility at the storage conditions. So what this plot shows is a fraction of SBD stable after 6 to 13 weeks. And what we use as an indicator of relative mobility is the TG minus the T, minus the storage temperature. And so the number in the bar chart is the number of compounds that we've seen here at Bend Research. And so what we can see here is that when we are greater than 30 degrees above the TG of an amorphous solid dispersion, 100% of the time, for 119 compounds, we are stable after 6 to 13 weeks. And so from this, we have a rule of thumb that the TG of the amorphous solid dispersion should be about 20 to 30 degrees above the storage temperature. And so before we make our formulations, we will use the Gordon-Taylor equation to predict the glass transition temperature of the dispersion. And what we can do is we take the Gordon-Taylor equation and we use some simplifying assumptions and end up with the Fox equation. And we can use this and plot polymer and API predicted TG on the graph on the left. And so what we can see is the TG lower limit line drawn in there would be the line, assuming the TG of the API is 30 degrees, it would be the line that we would like the dispersion to be above. And so in this particular case, we can see that the drug loading we can achieve a very high drug loading for almost all of these polymers. However, this isn't the whole case because a lot of these polymers will take up water. And so what this shows is water sorption and glass transition temperature for selected dispersion polymers. And they take up water via hydrogen bonding because of the amorphous structure of the polymer. And different polymers take up different amounts of water. Well, water has a TG that is minus 138 C, and so you get a plasticization effect. And this lowers the TG of the polymer in the solid dispersion. So what we can do is we can take the equation that we used on the previous slide, the Fox equation, and we can take what we know about a TG versus RH of the polymers, and we can map drug loading and relative humidity. We can map glass transition temperature as a function of drug loading and relative humidity. And these plots really help us understand the potential effect of RH and drug loading on the physical stability of the solid dispersion. Now this impacts the shelf life and may indicate if special packaging may be necessary. So in these plots, what we can see is that the drug and the PVP, although they have a higher TG when there is no water present, once the water is present, the TG dramatically decreases, whereas the drug in the HPMCAS has a wider formulation operating space. And the TG equals 60 degrees C line is there to show, kind of as a marker to show the difference. Assuming the drug has a TG of 30, this would be 30 degrees above the drug TG. The next kind of question we ask is the API chemically compatible with the dispersion polymer? And this really is on a per API case, and so I won't go much into it, but we will do kind of force degradation studies and determine decomposition kinetics, and then model that to determine how we can minimize the effect within the extruder. Finally, the last question we asked during our formulation selection criteria is, does the polymer enhance and sustain the solubility of the API to meet the target PK profile? And we will answer this question by using a in vitro gastric buffer, intestinal buffer transfer microcentrifuge dissolution test. And this is one of our dissolution tests where we expose the solid dispersion to gastric buffer at pH 2, and then we expose it to intestinal buffer at pH 6.5. And what we're looking for here is we're looking for solubility enhancement and sustainment. So looking at this, we can see the crystalline drug is at the bottom. And two things to note. The 25% drug and HPMCAS M grade HME and the HPMCP HME both go to a very high soluble concentration and then crash down to crystalline drug. Whereas the 25% H grade and 15% H grade HME solid dispersions go to that high solubility and sustain. So finally, once we have our formulation selection operating space kind of mapped out, then we will turn to our HME process operating space. And here the goal is to obtain a single-phase amorphous solid dispersion. And so we talked about the binary phase diagram, and we do use this during this when we define our operating space. And we also determine if we need to use any processing aids and how this may affect that phase diagram. So whether we need to use a plasticizer or any other thing to aid in dissolution. We will also determine the processing constraints based on the degradation of the API in the polymer. So what this shows is this is a plot that quantifies an operating space where a homogeneous solid dispersion with acceptable or no degradation of the API or polymer is obtained. And really, this is an iterative process throughout the development and scale-up. In this particular plot, this assumes a fixed L over D, length over diameter, and screw design of the extruder. Now, there are lots of ways to do these plots, and this is just one way. Here what we do is we start by defining the top dashed line is the polymer or API damage line. So it's our operating temperature we have to stay below before we get significant degradation of the polymer or API. And then bisecting that line is the complete dispersion line. And so we need, it kind of defines our lower temperature where we can get a complete dispersion. And the lines on the side of the mean residence time are the equipment operating lines, meaning that for our fastest and slowest feed rate. And this is all versus mean residence time. And so we have a very good way of measuring the mean residence time for a particular screw design and extruder setup. And so we define the mean residence time is when 50% of a tracer has passed through the extruder. And this is the example of a tracer plot is in the plot on the left. And the variance is also important, and this is a measure of the axial mixing in the extruder. And so the variance is how wide that tracer line is, or the tracer distribution is. And this is important, especially if we have heat-sensitive material, or like we were talking about earlier, if there was degradation of the API or the polymer. And the way we do this is as we're processing, we will add a 250 milligram pulse of titanium dioxide, and we will monitor the extrudate with a Raman spectrophotometer, and this will give us our residence time distribution. And now for a fixed L over D and screw design, feed rate has the largest impact on residence time and the residence time span. And so decreasing the feed rate increases ramastication and therefore increases the mean residence time and span. Decreasing the process temperature decreases the mean residence time due to a decrease in the polymer's modulus. However, mostly residence time is independent of viscosity. And to note, not shown here, but there is a minor effect on screw speed. But all in all, residence time is very sensitive to feed rate. Finally, we have our initial range finding and development scale runs. And this goes to help further define our process operating space. And we can tailor this to the amount of API that's available for this work. So this can be simple parametric studies, or we can do a design of experiments. We'll typically do a screening design of experiments. And the way we do this is we will set up our experiment, and we will set it up so that we can use the continuous variables, which is feed rate, screw speed, and barrel temperature. And we will look to see if the extrudate is translucent. And if the extrudate is translucent, we will do our first tier testing on it, which is PXRD and SEM to determine if it's amorphous, polar light microscopy or hot stage microscopy, DSC to look for phase separation, and in some cases vibrational spectroscopy or solid state NMR, and this has to do with the melt homogeneity. Our second tier testing includes dissolution and free drug measurements and potency and purity. Now, if the polymer or API has degraded or the polymer is browning, we will go back to the initial parametric study or screening design of experiments, and we will decrease the amount of specific energy, which is the feed rate and the screw speed. We will decrease barrel temperatures or decrease the mixing. And decreasing the mixing means changing the mixing sections within the screw design, changing the kneading block sections, and or the length over diameter of the extruder. However, if the initial extrudate is not translucent, or if the tests indicate that it's not homogeneous, meaning that it's phase separated, crystalline, or has poor physical stability, we will increase the specific energy, increase the temperature, increase the mixing. And we will do these in a succinct fashion that allows us to determine our operating space. And now typically on our 18 millimeter light stress extruder, for continuous variables, we use 5 minutes to come to steady state, which uses 50 grams to 200 grams of blend, and then 5 minutes per variable for continuous variables. Finally, what we'll talk about is process scale-up. And okay, so this slide shows how we will scale a process from our development to our pilot scale. And we'll typically do this by looking at three numbers: the power number, the pressure number, and the throughput number. And this is fairly straightforward because we are going from a light stress 18 millimeter twin screw extruder to a light stress 27 millimeter twin screw extruder. For the power number, for equal viscosities and equal length over diameters at both scales, and comparing power to volume ratios, to equate the power number, all we need to do is match screw speed. For pressure number, again, for equal pressure numbers at two scales, and then for equal viscosity and L over D at both scales, all we have to do is match screw speed. And then finally, the throughput number is based on the volume of the 27 millimeter extruder versus the 18 millimeter extruder. And so what we'll have to do here is change the feed rate by 3.5-fold. So the 27 millimeter has a throughput nominally 3.5 times what is run on the 18 millimeter. And again, this is specifically related to scaling from Venn Research's 18 millimeter light stress twin screw extruder to the light stress 27 millimeter twin screw extruder. And so that ends our presentation for hot melt extrusion of amorphous solid dispersions for bioavailability enhancement. And if you have any questions, please contact us at the email below.
So that concludes our presentation. Thanks very much, Aaron. That was excellent. I see that we have a few questions, and we have about 5 minutes to answer them, so we'll get through what we can, and then if we don't answer your specific question, we'll follow up directly with you over email. Thanks again for your questions. So let's get started. So Aaron, the first question is HME feasible below the melt point of an API?
So thank you, David. That's a great question. And yes, HME is feasible below the melt point of an API. At this point, if the API and the polymer are miscible at the processing temperature, this becomes a mixing problem and a dissolution problem. So what we need to do is figure out how does the API dissolve into the molten polymer, and is there going to be any particle size effect on that dissolution rate.
Okay, great. Another question, how much API is needed to do the initial assessment at a lab scale?
So that's a great question, David. So the amount of API to do the initial assessment is anywhere, can be as low as a gram. And this is based on doing some very small scale spray drying and then doing some thermal characterization by DSC and some in vitro dissolution testing.
Great, thanks. The next question is, do you have any examples of the potential used APIs and with which polymer? So maybe the question's around what type of API are appropriate for which polymer?
Right, so in general, what we'll do is we have to characterize the API, and as seen in the presentation, we'll look at the solubility parameters, and we'll look at, a lot of times we'll look at drug-polymer interactions using spectroscopic techniques, and typically the more interaction you see, the polymer will work to stabilize the drug in the amorphous form better.
And maybe something around temperature compatibilities and things like that.
Yes.
You know, relative to the thermal stability of the polymer versus the melting point of the drug, things like that.
Yep.
Great.
Thanks, David.
If necessary for processing, how do you choose a plasticizer?
That's a great question. Thanks, David. So typically, we'll start with a binary system, and that's just polymer and API. And the API often can act as a plasticizer, and this is often enough to reduce the melt viscosity to allow processing. However, if we do need a plasticizer, what we'll do is we'll start our selection based on matching solubility parameters of the plasticizer with the polymer, just to ensure that this doesn't change the phase diagram between the drug and the polymer. We will also make sure that it lowers the melt viscosity enough to allow processing, and check to see if the plasticizer has any impact on dissolution. However, there is a balance, because usually a plasticizer will make the material less brittle, and therefore it may impact downstream processing.
Great. The next question, is there a way to quantify the physical stability of a kinetically stabilized amorphous solid dispersion to predict shelf life?
Oh, thanks, David. That's a great question as well. So yeah, Bend Research has many techniques, analytical techniques we use to do this, and in particular we'll use a thermal activity monitor, and we'll be able to take the results from that, quantify them, and put them in some of the models that we have developed. And these models are used to predict shelf life.
So sort of measuring crystallization kinetics at elevated mobility conditions and using that to extrapolate to relevant conditions for pharmaceutical storage.
Yep. Well said, David.
All right. For our final question, is Bend Research experience with dosage form development using the amorphous solid dispersion intermediate?
Yeah, so we are very experienced in formulating amorphous solid dispersions into immediate release tablets and modified release tablets as well. We're also very experienced in formulating these into capsules.
Okay, and That was such a succinct answer. We'll do one more. Is a similar scale-up strategy employed when going to commercial manufacturing?
So yeah, that's a great question. So in general, the extruder size is dependent on the outer diameter of the screw. And if we're scaling from a similar extruder, meaning we're scaling from a light stritz 18 to a 27 to a 50 millimeter, in general, the free volume of the extruder, the ratio of the free volume to the size of the extruder is the same. So it's very easy to scale up, and the process is very well known. If we are scaling from a different brand of extruder where the free volume is different, then we will take a look at specific energy, including the mechanical energy and the thermal energy. We'll also look at the residence time to make sure that we're matching not only the different energies we're putting into the system, but the time that the amorphous solid dispersion sees those energies as well.
Okay, great. Thanks again, Aaron. Excellent presentation. I think we all learned a lot. So if we didn't get to your question, we'll follow up directly over email, as I said. And if you have any additional follow-up questions, please feel free to contact us directly via email. Again, this is a webinar in a series of webinars around enabling technologies for bioavailability enhancement. The next webinar in the series will discuss and address lipid and liquid fill technologies for enhancing bioavailability. So please check our website for details and to register. On behalf of Capsugel DFS, myself and Aaron, I'd like to thank you all for joining us today. Please fill out the survey that will pop up on your screens as soon as the webinar is over, and your feedback will be incorporated and help us to make these information webinars more relevant to the type of research that everyone's doing. So thanks again for your time and attendance today.